worked in the KM Neuroscience lab. Now, what does this lab do? The KM lab studies temporal lobe epilepsy and is developing an optogenetic intervention method to stop seizures. Epilepsy is a series of recurring seizures and can be either genetic or acquired later due to trauma. Temporal lobe epilepsy is the most common type of epilepsy in adult humans. Seizures in the hippocampus can be caused by a hyperactivity of cells in the hippocampus. This means that we can induce seizures in mouse models by injecting a neurotransmitter called kainate into the hippocampus. Inducing seizures in mice is the first step of many in stopping seizures. Here is the step-by-step process of optogenetic cessation of seizures. First, we induce temporal lobe epilepsy in transgenic mice. I worked with Kara, the junior scientist in the lab, and Zach, a graduate student, to perform these steps. We perform surgery on mice to inject kainate into their hippocampus. Here is a coronal section of a mouse brain. The inset is the mouse's hippocampus. When kainate is injected into the hippocampus, it causes sodium channels to open. This causes an influx in cations, which depolarizes the cells. Eventually, a whole network of cells fire too many action potentials, which leads to status epilepticus, or the initial seizure after trauma. Then the mouse develops chronic epilepsy, or reoccurring seizures. Pictured on top left is the mouse brain that I injected Florigold into. This is what it looks like under the microscope. Directly below is the same slice, but under light that illuminates where the Florigold is. The picture on the right shows a slide with 5 mouse brain slices under the microscope. The second step in stopping seizures in mice is to implant optrodes and electrodes into the mouse brain after having induced epilepsy. The optrode shines light on specific cells, while the electrode reads cell activity. The third step is to deliver light to the cerebellum, where we've implanted the optrode. Then we can analyze the data with the EEG and score the mouse's seizural behavior. The fifth step is to process these results. Here are the results for one set of data that I analyzed. The graph on the next slide will show the percent of seizures that stopped within 5 seconds of light intervention. It turned out that about 85% of these seizures stopped within 5 seconds after the light trigger. Finally, we want to ask the question of whether stopping seizures has a positive cognitive outcome. To answer this question, we run behavioral experiments. I worked with Zach, a graduate student in the lab, to set up 3 behavioral experiments. Zach is pictured on the right of the screen, as well as in the next slide with me. We wanted to test improved cognitive performance in 3 behavioral experiments, the first one being the novel object recognition task, the second one is the object location memory task, and the third one is a modified Morse water maze. The lab will run these experiments on 3 groups of mice. One group of mice will receive the light intervention, and the cells will respond to the light because they will have been bred to express a particular light-sensitive opsin. A second group of mice will also express that light-sensitive opsin, but will not receive the light intervention. The third group of mice will receive the light intervention, but will not the cells will not react to it because they're non-transgenic mice. By running these behavioral experiments, the lab hopes to locate which cells to stimulate with light in order to most effectively stop the seizures. We'll also be able to assess cognitive costs of the treatment in non-epileptic mice. Finally, we'll be able to compare the timing of light intervention and determine whether an on-demand or non-on-demand approach works better. Here are a few pictures of me running a pilot study. I ran the object's location memory task. So there you have it, the step-by-step process of optogenetic cessation of seizures in mice. Thanks to Esther Croak-Magnuson, the members of the lab, and the University of Minnesota, as well as the CC Career Center, for making this wonderful summer internship experience possible.